Okay. Good morning and good afternoon to all of you, depending on whichever part of the world you are in. I am Sujit Nair, and I am currently serving as the Director of Technical Operations with Spectratix Pharma, which is based in Maharashtra, India. Spectratix Pharma is a new generation pharmaceutical industry which is involved in product development and marketing of pharmaceutical products. Through our academic arm, that is PharmaGyan Academy, we try to conduct lot of training programs, both for industry and academia. We run a large number of programs with the aim so as to have a good quality manpower for the industry. So one such series, I'm sure many of you are aware of this, is the Spectratix Knowledge Series, which is actually a weekly session wherein we invite different experts from both industry and academia to share their expertise and knowledge for the benefit of students, freshers, researchers, and also industry personnel who look to upgrade themselves or to gain or improve their knowledge. For this, we have also created a community. I'm very sure most of you are already part of that. This community actually serves as a platform to connect and also support each other's growth. Uh, in continuing with this lecture series or the knowledge series, today's talk has been titled as Nature to Clinic, translating bioactive compounds into personalized therapeutic solutions. And we are very happy to have a resource person as Dr. Vinaya K. She is currently the head of operations and R&D at WellGenome Biotechnology, which is based in Cochin, Kerala. It's a precision health company focused on nutrigenomics and lifestyle-based wellness solutions. She has more than 10 years of experience spanning both academia and industry. She specializes in the development of high-value, clinically backed and patentable nutraceutical and probiotic formulations. Her work uniquely integrates both traditional knowledge, marine bioresources, and modern biotechnology to address our lifestyle diseases through evidence-based natural health products. She holds a PhD in biotechnology from Central Marine Fisheries Research Institute, registered under the Cochin University of Science and Technology, where she pioneered research on marine macroalgal symbiotic bacteria for therapeutic and nutraceutical applications. She has held leadership positions in multiple biotechnology companies, contributing to innovations in product development, clinical and preclinical validation, IP strategy, scientific communication. Her research and development contributions include novel formulations like pre and probiotic formulations, and with both national and international patent filings and one granted patent. She has authored over 15 peer-reviewed publications in several reputed journals, and she also serves as a speaker to numerous national and international conferences. So I request the audience to be very interactive. I keep saying this thing. Also, make proper utilization of the resource person who these sessions. Also, one reminder that certificates will be issued only to participants who attend the full session. That is minimum of 45 minutes. So with that, I thank Vinaya ma'am for accepting our invitation. It's all over to you, ma'am.
Yeah. So good afternoon, everyone. Hope I'm audible.
Yes, ma'am. Audible. Just share your
So
screen.
once again. So thank you very much for the lovely introduction. I'll just share my PPT here. Hope it's clear.
Not yet.
It has not come yet.
No.
For me, it's showing it's being shared.
Can you reshare, please?
Yeah, I'm trying to stop it, but I'll just leave and rejoin then.
Fine, fine. Yes, now it's clear.
No?
Yes.
Yeah,
It
I'm
was.
audible now.
Yes, yes, audible.
It was muted. Sorry. Yeah. Yes.
Fine.
So once again, good afternoon, everyone. I hope all of you are doing well. I just wanted to make this session a little bit interactive because this afternoon, we might be feeling a little bit of sleepy. So I just want to make sure that you all are there with me. Okay. Please say anything in. I hope you all are there.
Yes, they are all there. They're dropping the messages.
Yes, madam, very much.
Oh, okay, fine. So, today I'm going to speak about, you can see the title, From Nature to Clinic, Translating Natural Bioactive Compounds into Personalized Therapeutic Solution. So I can say in a simple line, this entire webinar is about how a molecule that originates in nature eventually becomes a therapy for a specific or a particular person or a patient. Before starting, let me ask one quick question. How many of you have taken a supplement or a nutraceutical or a herbal product at least once in your life? At least you will be familiar with the term, right? Nutraceuticals, yeah. Yeah, one way or the other, yeah, we'll be familiar with the nutraceuticals, nutrition supplements, or natural product. We can see that in our kitchen too, right? Daily we are using it. The common one, the common example, everyone citing as a natural product or a nutraceutical, which one is that?
They are mentioned in the chat box, spirulina and ashwagandha capsules.
Okay, a simple one I can say turmeric.
Haldi.
All those comes under nutraceutical or natural product, that's true. It has many bioactive effects also. So say, for example, we can say turmeric, probiotics, ashwagandha, spirulina. Yes, all these are used for used as anti-inflammatory agents for gut health, for stress management. This fish oil we call omega-3 fatty acid, that is used for cardiovascular health. So in one way or the other, we all are familiar with, either we are taking it or we might have at least heard about that, at least seen that. And one more question, did it work the same way for everyone? Usually we know that the one drug or one supplement, it doesn't work in same way for everyone, right? The answer is no. Because for one person might say that this worked wonderfully for me. The same supplement or the ingredient that might not work same way for another person, and sometimes that might be allergic to. And another what we used to hear is two people will be following same diet, but one loses weight and improves metabolic markers while the other does not. So that is the entry point into today's subject. Natural compounds cannot be understood only in a general way. Increasingly, they must be understood in a personalized way. So the entire discussion today will move in 5 logical steps. First one, we will understand the evolutionary origin of bioactive compounds. Secondly, we will look into their mode of action, the mechanism by which it is acting at molecular levels and systems level. Then we will look into how the modern discovery has shifted from conventional, that trial and error methods, to multi-omics and computational science. Then the fourth one, we will discuss the translation barriers. Though we know that it has a potential or it has a good effect in vitro, still it is not coming out as a drug. Why it is so? What is the challenge? So that also we'll discuss about. Then finally, the fifth one, we'll understand the personalization and advanced delivery, the delivery methods. So this flow is like this, from the molecules to the mechanism of action, then its validation and how it can be delivered effectively, and in a patient point of view, how it can be personalized. It might be sounds big, but the logic is actually very clean, and we are simply answering 3 practical questions, like why nature is so powerful, why many natural compounds fail in the drug development process. As I mentioned before, it has very much, all of them are having very good activity, but still there is no a single, no, I cannot say as such, as a natural product as such is not becoming a drug. And what is the challenge? And how we can make them work for the right person rather than for an imaginary average patient. Like one size fits all, that leaving that concept, how it can be personalized. So with that roadmap in mind, let's begin at the beginning. Why nature is such a powerful drug designer? See, imagine this nature as the original pharmacy. So again, I'm asking one question. If you had to choose between nature and synthetic chemistry, which is the better drug designer?
Answer is being given as natural.
Okay, great. Okay. Yes, but many people instinctively they will think that the modern medicine or the modern way of making drug development or making drug, that is the best way. But the thing is, I'm not arguing about that. The thing is that when you think carefully, nature has been doing molecular design for millions of years. Again, just imagine these plants, microorganisms, fungus, and these marine organisms. They cannot run away from predators, right? They cannot relocate easily. They cannot physically defend themselves from away from their predators or competitors like animals do. So what do? They are sessile. They cannot move, so they have to produce some. They have to make some defense mechanisms. So what they do, they produce some defensive molecules, some chemicals, some toxic molecules, some signaling molecules, competitive molecules, and all. Generally, what we call them as secondary metabolites. And all these secondary metabolites are known as natural bioactive compounds. They are different types, they have different targets, and we'll see how it acts, what are all they, and how it can be used. So generally, these compounds interact with proteins, membranes, nucleic acids, enzymes, receptors, and affect the entire cellular metabolic pathways. In other words, we can say this living systems evolved chemistry as survival technology. That is why natural products are often structurally rich and biologically meaningful. They are not random molecules. They are function-shaped molecules defined by evolutionary pressure. This is one reason natural compounds are structurally complex and they have multiple targets. They are not like drugs, synthetic drugs. They have multiple targets and strong bioactivity too. And the surprising thing is that more than 60% of the approved anti-cancer drugs and 75% of anti-infective agents, the origin of them are natural. It's like either they are derived from natural products or they are inspired from natural products. That means nature is not a side story in pharmacology. It is one of the foundations of pharmacology. Okay. You might have heard about this penicillin, right? What is the story behind penicillin? Whether it was an accidental discovery or that was a very well planned discovery.
You mentioned accidental.
Yeah. So that we call serendipity, right? That was an accidental discovery. So what is the story behind that? Who was the inventor? Yeah. So Alexander Fleming, he was growing Staphylococcus plates in his lab, and during one vacation, he kept it as such. And when he came back, one of the plate was contaminated with mold, Penicillium notatum, one fungus called Penicillium notatum. And he observed that nearby area, near this mold, there was no bacterial growth. That means there was halozones. So what he did, he cultured the mold growth, the fungus he cultured the fungus, then tested it on a infected mice and found it selectively killed the bacteria. That Staphylococcus that was killed by this particular metabolite or produced by this fungus by inhibiting cell wall peptidoglycan cross-linking by beta-lactam ring binding to transpeptidase enzymes. So that was not affecting the mammalian cells. That was the interesting part. It was killing the bacterium, but it was not affecting the mammalian cells. And that mistake, that changed the world. The first antibiotic, penicillin, came and that saved millions in World War II. So the lesson number one, what we have to learn here is that nature is not an alternative medicine. Nature is a foundational medicine. So one by one, we can see the bioactive compounds produced by plants, marine organisms, and microbial organisms. Plants, as I said before, the bioactive compounds can be categorized in different ways. So the plants generally produce alkaloids, flavonoids, terpenoids, tannins, phenolic acids, and all. Say for example, alkaloids. Morphine, you might have heard about, no? Morphine. So that is a neurotransmitter. So what this alkaloid that interacts with this neurotransmitter and ion channels. Similarly, quinine. So quinine is an anti-malarial drug that is also an alkaloid. Another one is flavonoids. Flavonoids generally found in fruits and all. They are rich in antioxidants. It also has several other bioactivities, but its radical scavenging activity is very high. Another type of compound is terpenoids, like menthol and all. Then what they do, they often interact with membranes or enzyme systems and may show antimicrobial, antiparasitic, anti-inflammatory, or anti-cancer activities. Another type of compound is tannins. They are kind of polyphenolic compounds that can bind to proteins, that can influence enzymes like microbial adhesion, and the same like flavonoids. It can control the oxidative stress too. And its taste is very familiar, like the taste of red wine you might be knowing. So that is due to this presence of these tannins. Red wine, pomegranate, that's for some bitter sour taste is there, no? So that is due to this tannins. So another polyphenol compound, if I say, aspirin, that NSAID drug, we are using aspirin.
Yes, it's been mentioned.
Okay, whether it is a natural origin or a synthetic one.
Vaibhav has mentioned as salicylic acid.
Yeah, it's acetyl salicylic acid. So I'm asking whether it is synthetically made or whether it is having some natural origin.
Natural, it is mentioned.
Okay, so so you know about that. So I'm not going deep into that. The means how it has been made. When it comes to marine organisms, as I mentioned before, especially the natural products, as I mentioned, they are very strong. They are unique. They are structurally complex too. When it comes to marine organism, it is much more deep in that case because they are exposed to extreme environmental conditions. Sometimes they will be in a very dark condition. There will not be sunlight at all, and very cold condition. Sometimes there will be thermal vents. They will be living in thermal vents. Then in intertidal area, they will be exposed to water. Sometimes they'll be under the water. Sometimes they'll be exposed to sun. So all these extreme conditions they have to survive. And then the second thing is whatever they produce, whichever chemicals they produce for defense or communication, they have to produce in either in bulk quantity or in very high concentration because it is getting diluted in water. And the other thing is it should not affect other organisms. If it is targeting their predator, it should affect that organism only. So that is very target specific too. And the most interesting fact is that the marine bioactive compounds, there is no alternative. There is no counterpart in terrestrial organisms. So if we are dividing or categorizing this marine derived bioactives, we can say peptides, polyketides, alkaloids, macrolides, and nucleoside analogs. So you might have heard about the drug cytarabine. That is an anti-cancer drug used to treat leukemia. That is one of the natural marine bioactive compound approved by FDA. Currently used to treat this leukemia. So its mode of action, when it comes to its mode of action, it's a pyrimidine analog that gets incorporated into DNA during that replication, and that inhibits the DNA polymerase, thereby halting DNA synthesis in rapidly dividing cells in cancer cells. So that is one of the examples for marine bioactive compounds. And one of the recent, many of the recent researchers are showing that previously it was observed that marine macroorganisms are most of the bioactive compound producers, but in many recent researches, it is showing like their symbiotic microorganisms. In most of the cases, their symbiotic microorganisms are the producers. So that means the microbes, they are the richest sources of bioactive compounds. So previously we have seen penicillin from a particular fungus, Penicillium notatum. So similarly, several other antibacterial, antimicrobial, anti-inflammatory, anti-cancer bioactive compounds are produced by these microorganisms. So we have seen different sources, different types, and we know that all these bioactive compounds are having potential bioactivities. But then the question comes, how exactly do these molecules work inside our system? We have to understand what is the mechanism, how it is being absorbed in the body, and how it is doing its work there. Generally, what we do when we develop a drug, we have a system already non-system, like one drug, one target. We used to design in such a way, isn't it? So that's what we usually learn. But the thing is, that is correct. But in case of natural products, they are not single targeted. They have multiple targets. So the drug or the molecule or the compound is powerful, but it's very difficult to learn and make it as a drug. That is one of the challenge what we face now. So when we think about this mode of action, when I'm taking first thing as cellular pathway modulation. Considers NF-kappa B. So again, I'm coming to that familiar example, curcumin, that is a active compound in turmeric. So what it does, this NF-kappa B, we know that one of the central transcription system in inflammation. So under inflammatory stimuli, what happens? The I-kappa B kinase complex phosphorylates I-kappa B, which marks it for degradation. Once I-kappa B is removed, what happens? NF-kappa B translocates into the nucleus and activates a gene for inflammatory mediators like TNF-alpha, IL-6, cyclooxygenase, like COX-2 and all. What this curcumin does, it can inhibit this pathway. So in simple terms, this curcumin suppresses this activation of I-kappa B kinase complex. So this I-kappa B degradation is reduced. What happened? NF-kappa B will not go to the nucleus. It will remain sequestered in the cytoplasm, and the inflammatory gene transcription is decreased. Thereby, the important point is that the compound is not just blocking one cytokine, it is modulating the regulatory architecture of inflammation. That means instead of blocking one signal, they regulate the entire system. And that's how this curcumin is acting as an anti-inflammatory agent. That is one of the mode of action mechanism by which the natural bioactive is working. Another thing is epigenetic modification or epigenetic regulation, we can say. These bioactive compounds can influence gene expression without changing the DNA sequence as such. So what do they do? we can call it as reprogramming biology. So it's just reprogramming it. This DNMTs, no, DNA methyl transferases, they add methyl groups to DNA, especially in the CPG islands. So that leads to gene silencing. Some bioactive compounds like curcumin or resveratrol, they can inhibit this DNMT activity and reduce the pathological hypomethylation, thereby helping reactive silence tumor suppressor genes. And that means it will reactivate this that particular gene. So we are not just targeting the proteins; we are also modifying gene expression landscape. We are not doing anything with the sequence, but the external environment, how the expression is modified, how the expression is regulated. I mean, the bioactive compound is focusing on the gene expression. And the third method is immune modulation. So as I mentioned before, the anti-inflammatory pathway, if you're taking this curcumin, they can regulate the cytokines like IL-6, TNF-alpha, and they can reduce the chronic inflammatory tone. That will affect the macrophage polarization and enhance the adaptive immune signaling, including T cell function. That makes them especially relevant in chronic inflammation, metabolic disease, cancer supportive strategies, and some immune-mediated conditions. So when we say this natural compound or natural bioactive, we do not simply mean that it exists in a plant or extract. We mean that it interacts with living systems in a measurable, mechanistically meaningful way. It may alter signaling pathway, transcription factors, as I mentioned before, oxidative stress balance, epigenetic regulation, immune behavior, membrane dynamics, or mitochondrial function. So this system level behavior is both an advantage and a disadvantage. So the advantage is, as I said, the compound may influence several disease relevant processes at once, as it is multi-targeted. The challenge is that such compounds do not fit neatly into a simplistic single target framework. So we need better tools to study them, and that takes us directly into omics and modern discovery science. We know the conventional method of drug discovery, especially in the case of natural products. What we do, we'll identify one target, I mean a source. Say, for example, if you're identifying one plant source, we will take that, extract it, take the crude extract, then we'll go for bioactivity guided fractionation and purification. We may have different compounds with different bioactivities. That too might be in pure form. But still, sometimes at the end, the yield might be very less, or it will not be a stable molecules. Sometimes the activity, what we are expecting might not be there. So the entire process is waste, right? So that's a kind of trial and error method. Of course, it has strength, like it was capable of unexpected discoveries and sometimes identified valuable compounds without prior mechanistic assumptions, as like our serendipities. That also happens. But it also had major limitations, like, as we know, it's slow, of course, then expensive, mechanistically incomplete, and inefficient when the search space becomes too large. And we know that if we have to identify 1,000 or 10,000 compounds, how long it will take? It may take years, right? So there comes the relevance of these multi-omics approach, computational modeling, or high throughput screening. So when it comes to multi-omics, it covers genomics. Genomics helps us understand the genes, variants like SNPs and all, then biosynthetic clusters and host response patterns. So in two ways we can see the genomics of that particular host. We can study the same way the genomics of the source, either whether it is plant, microorganism, or marine organism. The genomics of that particular source also we can study and we can modify it. We can do some genetic engineering. And sometimes if we are getting a bioactive compound from a microorganism or a non-sustainable source, what we can do some genetic engineering and make it production. I mean, make it very scalable. And on the other hand, if we can see, we can study the genomics of the host. We can make personalized solutions, personalized medicines, personalized suggestions and advice for that for the specific person or the specific patient. Then comes transcriptomics. So what does that do? That shows how RNA expression changes after exposure to a particular natural bioactive compound. Then proteomics identify changes in protein abundance, modification, or interaction networks. Then metabolomics. So metabolomics will give you, metabolomics is giving the exact picture, like the current picture. And that captures the downstream chemical consequences, which is especially important because the phenotype often manifests metabolically. Say, for example, if you're saying there is a gene expression or there is a SNP, there is a polymorphism, there is a variation in the gene. How we can identify that biochemically or very easily in clinics? So we can do some biochemical tests. And if there is any change in the final product, where sometimes it can be enzyme change or hormone change. Say, for example, there is an insulin resistance. We can see that in biochemical reaction, right? So that's how it helps. Metabolomics helps us in identifying that particular person's current health condition. So when all of these are integrated, we no longer ask only, does this compound work? So we can ask like which gene, which protein, which pathway, and metabolites change, in which direction, in which patient subtype, under what biological context. So we can answer all these questions if you're integrating all these omics. So that is multi-omics effect. So that is a much more clinically useful question. Then comes high throughput screening. As I mentioned before, if you're going to screen 1,000 or 10,000 compounds together, this high throughput method will help us to do so. So at a time, we can identify, we can analyze thousands or 10,000s of extracts, fractions of purified molecules. They can be tested and they can be tested against different disease targets. So that will dramatically reduce the time required to move from crude biological interest to the prioritized potential drug candidates. Another method comes the computational modeling. Say for example, if you're saying molecular docking. So that will help us to estimate how strongly and in what orientation a compound may bind to a target protein. Then machine learning that can integrate large data sets and predict not only efficacy but also selectivity, toxicity, and responder patterns. So before we see the bioactive compound in pure form or in drug form, we can predict all these things, like whether it has activity, in which target it is getting bound, then whether it is toxic, whether it is having any side effect, how it is acting inside your body. All these things we can predict. And then if it is effective, then only you go for physical purification. So this is why modern discovery is no longer simply chemical screening. It is data-driven biological inference. So now we have all these things from the conventional methods to the advanced method, and we have active compounds. We know the activity, we know how to screen them, but still there is a gap. Most of the natural products are not becoming drug. They are still in the stage of herbal supplements. I mean, in the form of herbal supplements, nutraceuticals, or functional ingredients, we can say, but they are not becoming a drug molecule. What can be the reason? I mean, what can be the reasons?
Lipinski rule, they mentioned.
Okay. In simple way, what we can say, we are taking a medicine, and if it is not reaching to the place where it has to be reached, what is the use of that? We know that it has, say for example, if I'm taking again curcumin, it has anti-inflammatory activity. We know that. We have proved that in lab, I mean in vitro. And we are taking curcumin, say 1 tablespoon of curcumin, but it is not reaching to the target point. There is no use of taking it, right? That means most.
Yes. Sorry.
Yeah, please.
One comment is that it's not fulfilling the cost effectiveness and appropriate therapeutic effect.
Yeah, the therapeutic effect means it has activity in vitro, but it's not showing in vivo. So what is happening here? Where is the gap? Most of the natural bioactive compounds are poorly bioavailable. It's not available. I mean, it's not getting absorbed in your body properly, and that too, it's not reaching to the point. So there is no use if you're taking a very highly bioactive compound. It's not reaching to the point. So the biological success is not determined by potency alone. It also depends on whether the compound reaches the target, survives the metabolism, distributes properly, and interacts meaningfully in the patient's biological context. So this is where pharmacokinetics becomes essential. We know that absorption, distribution, metabolism, and excretion, that process. So a compound taken orally may have poor solubility, water solubility. Then it has low intestinal absorption, intestinal instability in the gut, extensive first pass metabolism in the liver, rapid clearance, or limited tissue penetration. Any of these can destroy clinical usefulness. So again, this curcumin, so it's biologically impressive. We know its in vitro effects. We can see that in lab, as well as many publications are there with the activity of curcumin, and it's proven too. And maybe in the preliminary clinical trials too sometimes. Mostly in preclinical models, it's not very strong in clinical trials because its oral bioavailability is very poor. I mean, curcumin alone, I'm telling, because it has very water solubility, very less poor water solubility, chemical instability, rapid conjugation. It's not stable at all, and low plasma persistence. So even though it is potent in lab or on paper, it may never reach sufficient therapeutic concentration at the relevant tissue site. So that is why the statement is important. A good molecule is useless without good delivery. So that's why I mentioned curcumin alone, because nowadays you can see nano curcumin, liposomal curcumin, so many modified curcumins are there. I'm just mentioning about curcumin as an example of bioactive compounds. So why we are going for this nano? Why this liposome? Why we have to modify it and how we are doing it? So we'll come to that point. So before that, I'll just explain what are all the challenges in translating this natural bioactive compounds into drug or a therapeutic agent. Then comes to pharmacodynamics. Like I was explaining about the pharmacokinetics, then comes the pharmacodynamics. So we know that different patients have different receptor expression, their enzyme activity, transport systems, microbiome composition, and inflammatory states. So the same dose does not guarantee the same response. So that also we have to optimize. Now comes the personalization, the important point, the exciting part of this presentation. Why the same supplement or natural compound works differently in different people. As I said before, we have heard about, same two people will be taking the same supplement, but for one it is affecting, for the another one it is not at all effective, or sometimes it might be allergic to. Same way the diet. So what is the major reason? And that is actually that reason is our keystone in making the drug or the molecule or the natural bioactive personalized too. So the answer is genetics. But now when we say genetics, it has environment, metabolism, and physiology too. Because as I said before, the genetics, the epigenetics, even if we are not altering the DNA sequence, we can alter its surrounding environment, the epigenome. That means the expression. So that expression is influenced by environment, metabolism, and physiology. So when genetics comes, in the case of personalization, all these terms comes together. So that is one of the important thing what we have to study when we learn about this personalization is nutrigenomics. So what is that? Nutrigenomics studies how the genome influences the way a person responds to nutrients and dietary bioactives, and vice versa. How a nutrient is affecting one person's genetic makeup. Okay. So if I'm saying one example, this MTHFR gene you might have heard about, no? Methyl tetrahydrofolate reductase gene, that can alter the folate metabolism. So that is changing methylation capacity and 1-carbon metabolism. Similarly, apolipoprotein E. So if we can identify this variation, if there is any mutation, the SNPs are already proven, already reported, identified, and proven. So we can identify that easily. If there is a mutation, we can identify that, and accordingly we can give the treatment, or accordingly we can manage that particular condition through natural molecules, supplements, or diet. Then pharmacogenomics. Simply, what is pharmacogenomics? How a drug molecule is interacting with a patient or person's body. Same way, how a person is reacting to a particular drug molecule. So that is also linked with this genetics. So if we can identify whether a person is allergic to a particular drug or it is not at all effective on him, if we can, if we can identify that beforehand itself, we can give a personalized treatment to that person. So simply, if you're saying taking this nutrigenomics, pharmacogenomics, all these together, we are moving away from one size fits all concept to the principle of the right compound for the right patient. That means from one size fits all to what is best for you. So that's what we are trying to find out through personalization. So that is a scientific basis of patient stratification. Once we identify the responders and non-responders more accurately, clinical trial design improves and therapeutic outcomes improve. So I was mentioning about this. I can continue,
Yes, ma'am. Yes, please.
Yeah. So I was mentioning about this bioavailability, then how we are you might have heard about this nano curcumin, liposomal curcumin and all. So what is the difference between this pure curcumin and nano curcumin? And why we are going for this nano curcumin? The answer for why is we have already seen that there is a poor bioavailability for this curcumin. That means generally if you're saying for the natural bioactive compounds, most of them are poorly bioavailable. So we have to make it bioavailable in different ways. The delivery, the drug delivery or the molecule delivery should be in such a way that it should be absorbed by the body and it should reach to the specific tissue target site. So we have to take the molecule from the site to the target site. So one of the method is making it as nano emulsions, or we are making liposomal curcumin, and thereby we are improving the pharmacokinetic profile. We are improving the bioavailability, we are improving the water solubility, aqueous solubility, and we are protecting the payload from degradation, improving its circulation time, and enabling tissue targeting. This is one of the example I have just mentioned about, how to improve the bioavailability. One of the methods that can be used to overcome the challenge of poor bioavailability. Similarly, keeping all these out, if you are thinking only about our ancient methods, like in India specifically, they're using different kind of spices, right? So if you're adding spices, we might not have done any kind of in vitro or in vivo studies, but our ancestors have said that if you're adding curcumin, you have to add a pinch of pepper also. If you're adding turmeric, you have to add a pinch of pepper also in that. On the other hand, they always add this turmeric into oil. All these simple tips, all these things are because it is not bioavailable, and when you add this pepper along with this turmeric, pepper is increasing its bioavailability. Means its stability time, it is not getting excreted from the body. Its metabolism, pepper is delaying the metabolism of curcumin, and more time it will be available in the body, and it can be absorbed. More concentration can be absorbed. So the bioavailability, it is increasing. So there are different ways like this, like nanotechnology. There are different other ways to deliver this natural bioactive compounds in better ways. So if we can overcome that challenge, that bioavailability challenge, in one way we can improve this bioactive compounds reaching into the therapeutic category. One of the example, if we are saying, if I'm saying this omega-3 fatty acids, EPA and DHA, FDA has approved this EPA and DHA as such. So when we connect this everything, when we connect everything in real clinical area, say for example, if you're taking oncology, cancer studies, this bioactive such as curcumin, resveratrol, they are studied very well because they have influenced apoptosis, angiogenesis, metastasis, and inflammatory signaling. The attraction in cancer biology is not that they are magical, but they, as I said, they are multi-targeted and they touch several hallmarks of cancer simultaneously. Hence, the future direction is the convergence of AI, multi-omics, regulatory science, and precision medicine. So, multi-omics integration allows us to map compound patient interaction landscapes in a more complete way by layering genomics, epigenomics, transcriptomics, proteomics, and metabolomics. Similarly, if you're taking this AI-driven discovery, you can predict novel scaffolds, optimize analogs, identify hidden response signatures, and match compounds to patient profiles at scale. So at the same time, regulatory frameworks from bodies like World Health Organization, FDA, and EMA, they are increasingly emphasizing standardization, phytochemical characterization, reproducibility, and rigorous safety evidence for botanical and natural product-based therapeutics. So the big picture is this: the next generation of natural medicine will not be vague, generic, or purely traditional based. It will be evidence-based, evidence-linked, science-backed, delivery-enabled, mechanism-aware, and patient-specific. So let me end with the central thought that medicine is moving from simply treating disease to understanding the individual, your gene, your metabolism, your inflammatory state, your absorption profile, your response pattern. Similarly, the bioactive compounds too. And once we understand the individual biology more deeply, we can choose interventions more intelligently. Thank you so much. So I'd like to take your questions now. And before that, let me add these 2 points too. I was mentioning about this different delivery methods to overcome the challenges of poor bioavailability. In our lab, in our company, what we do is we are also using this nano emulsions, nano emulsified nutraceuticals we are developing. Same way, all these nutraceuticals we are using in personalized way. That means we are identifying the genetic makeup of a particular person. Similarly, the microbiome, the gut microbiome profiling also. And through that, we are giving a personalized advice, suggestion, and therapy for the patients, for the person. I would like to invite you all for the upcoming international conference we are organizing during May 29 to 31st in Cochin. So hope we will meet soon. Yeah, thank you.
Thank you, ma'am. We have a few questions in the chat box. Can you see them?
Yeah, one second. I'll just take it. Yeah. So first, I think, sorry. How do inherent natural bacteria like plant genetics or climate harvest timing and extraction inconsistencies challenge the standardization of bioactive compounds for reproducible clinical dosing, and what analytical strategies could bridge the gap between marker-based testing and true bioactivity prediction? Okay, this is on the other hand, like the genomics, I mean the genetic pattern of the source we are identifying, right? In that way, you're asking. I mean the genetics, climate, harvest timing. So we are modifying the source. Am I correct?
I'm not getting the name. GJB, if you are there, you can directly ask.
Yeah, I mean to say that. Can you hear? Is it?
Yes.
Yeah, yeah. Audible.
Yes.
Hello.
Please. Yes, please continue.
I would like to say that first of all, bioactive compound should be standardized. It should be reproducible in the commercial angle. R&D, we can say that personally everything, but it should give a same impact, same effect. So how it will be predicted for the bioactivity of a reproducible way? Like if you are personalized also, we can consider like medicine also. And this is there are many variable factors are there, natural variabilities. So incorporating all the variabilities, because there is different timings if you extract any bioactive compound, you don't know that whether it is the same timing also. So how do you standardize that bioactive compound to produce for the same repeated to everybody? Just like we can say that in the medicine there is a standardization, or any phyto compound, some of them it is standardized. So how it is in this scenario? Do you get my point? No?
Okay. It's like dosage or something.
Yeah,
Actually,
clinical doses, because clinical only once you dose once, but it will give you the result. But when it is a part of a commercial way, repeated to everybody, it's same similar kind of metabolic disorder or whatsoever. So how you can consider the bioactivity prediction? True biotech marker compound testing, we can know that these are the you are getting the test. Okay.
oh, okay. In production way, there also like we have to identify certain markers. Like in each batch, whether there should be a marker.
But comparison with marker with the bioactive prediction, because the final bioactive prediction should be there. Whatever the end outcome, what we predict it should be reproducible. What and how it is nothing but by making a standardization. It will help us. So how it will be standardized? That's the question.
And according to my suggestion, if we have to get a sustained result, like a constant bioactivity result, one thing we can go for this genetic modification. I mean, genetically engineered organisms can be made to produce this compound. One thing. Second thing, we can analyze it. Actually, I have to look into it other ways.
Because analytic is a part of that. You have to prove that your product is okay. Justify it. Strategy should be in a sense that whatever the compound which we have prepared, it should
No, actually,
give the desired
if you're
effect.
saying about this formulation, there we can see it's when we're talking about ingredient. I mean, the nutraceutical ingredient during the production. Actually, we cannot. If you are taking it from the direct source, like say for example, if it is a plant source, during making it, when during extraction process or purification process, at that time we cannot. I don't know whether as such we can say a constant bioactivity prediction is possible there. When we make a formulation, at that time we can optimize it. The production we can optimize, the combination we can optimize, the process we can optimize, and we can make sure that in each batch you are getting the same bioactive products or the end products. That's how we can make it sure. But in the case of ingredients, I mean the extracts, if you need a constant result, one thing is we can go for this genetic engineering. The other way, I mean directly from the nature, I don't know how to make it uniform in each batch. But in formulation, we can do that.
There is a concept of quantum biology also, where part of it is within this.
You are asking what I understand. If I'm sorry if I'm wrong, you are asking about the strategies which can help the reduce this issues, right?
Because it is a variability is very high in natural.
Understood. So you are asking how you can reduce the variability, right?
Yeah. So that is a major challenges for a product used in the therapeutic. Because your clinical, when you do the clinical dosing, you will get a one result. When during the actual use, it may have a variability. So how we need to standardize that?
Means it will ultimately depend on the strategy that you are following, right?
Yeah,
Means
strategy.
you can follow, means you can optimize your conditions, isn't it, ma'am?
Yes. While extracting, that is the only way.
Because we can't control the variability in nature.
That we cannot predict. Because the natural source may vary, the climate, everything may vary. So that optimization.
Means you can
Yeah, that I have to look into. While making it as a final product, final formulation, at that time we can do.
Yes, exactly. Means when you are extracting also, if you are extracting, you can actually standardize your procedure so that you start getting consistent results. Means whether it is a selection of the raw material or something.
And same conditions we have to grow. During same seasons we have to harvest. All these things, if we are, that is a way of making it. But, yes, that optimized method we have to identify.
Thank you.
I guess you have answered this. What may be the epigenetic factors affecting drug absorption from person to person?
What may be the epigenetic factors affecting drug absorption from person to person? It's of course the expression, that single one we cannot say. How the gene expression is modified. whether it is silenced or, I mean, whether it is upregulated or downregulated, that depends on the person. As I said before, the genetics, this epigenome or epigenetics or genetics, we cannot say alone. It is also influenced by its habits, his surrounding environments, his food habits, then the toxins, what he's exposed to. So all these affects the gene expression. So that we have to identify and that we have to regulate.
Also the lifestyle.
Yeah, the habits.
Okay, I guess those are the...
I'm sorry, I just forgot to mention the particle size. When we are making it nano, actually we are improving and we are reducing the particle size. That way, yes, the absorption is delayed. What are the major degradation pathways? Hydrolysis, oxidation, photolysis, both active compounds, and how do they affect therapeutic efficacy? Major degradation. For that, if I'm explaining, do you foresee future where personalized plant-based natural therapeutics could replace synthetic drugs for chronic diseases like diabetes or cancer? Okay, the first one, what are the major degradation pathway? If you say yes, same, hydrolysis, oxidation, photodegradation, yeah, photolysis, thermal degradation, and it can be enzymatic degradation. Also, as I mentioned, microbial and storage related deterioration. Means if there is a contamination or something like that, that is also affecting the degradation. I mean, that is also one of the degradation reason. So, in practical, if we say the resveratrol and curcumin, it's one of the challenge of using all these bioactive compounds is it's instead, I mean, it's not stable because it is getting metabolized easily, and degradation rather that means it is getting degraded into less active forms. So, and how do they affect therapeutic efficacy? Like it is not reaching the bioactive compound which is active in the active moiety, which is not reaching to the target site, the target tissue site at a proper concentration, at a required concentration, because either it is getting metabolized or it is getting degraded, or the molecule is unstable. So it is not reaching to the target site. So that activity is not happening there. Bioactivity, which we are expecting, that is not happening at all.
Means there is a reduced dose delivery.
Yeah, there will be a required dose to have that activity, so that particular amount will not reach, that is not reaching there, either because of this poor bioavailability or rapid metabolism, or this degradation, rapid degradation. So that is of course reducing the therapeutic efficacy. And do you foresee a future where personalized plant-based or natural therapeutics could replace synthetic drugs for chronic diseases like diabetes or cancer? there are nutraceutical formulations. I cannot say a particular natural drug to replace the synthetic drugs, but still there are alternative sources, like several compounds have been shown their anti-diabetic activity and anti-cancer activity. But as I said, it's not becoming drug because of all these challenges. But on the other hand, if you are improving its delivery method, like this, making it as nano formulations or liposomal formulations or something like that, we can introduce nutraceutical formulations to replace the synthetic drugs. That too, of course, personalized. But I was getting questions like whether it can be used in emergency conditions. But see, as I said, these nutraceutical formulations or natural products, they are still it's not in a pure form. What we do is we will purify and identify its structure. I mean the bioactive molecule or the bioactive moiety. Then either we have to go for a green synthesis method for only the active moiety. That might not be easy to purify in that way. We have to go for synthesis. Second thing, say for example, if you're saying this curcuminoids, there are 3. Curcuminoid is not a pure one. It's 3 curcuminoids. Similar with this pure compound, if it is a natural one, we cannot still say that it is very pure. A drug molecule should be, it should be small too. Almost all natural compounds are big, macromolecules. So there are several challenges. So it's very difficult to become a drug that can replace synthetic drugs, but still that can be used. The formulations or the bioactive compound supplements, like natural supplements or herbal supplements, can be used to treat or manage these chronic conditions, disease conditions. But still not an emergency drug, not as an emergency drug.
Yes, they can be used simultaneously.
Yeah, like adjuvant, it can be used as an adjuvant, of course. It will increase the effect, improve the effect. Second thing, as we said, if you are improving the food habit and all, no. Similarly, if you are taking this supplements regularly, we can manage that condition.
Yes. Okay, ma'am. Thank you so much, Dr. Vinaya. I guess those were the questions. You have dealt with the topic in depth, and we will be having you more often to discuss these topics in detail also.
Sure, thank you.
Okay, thank you so much. As far as the certificate question is concerned, we will be providing the certificates, and whoever have attended, they can directly share their ZOA ID to me. We will check the attendance and then start issuing the certificates. Thank you so much, Dr. Vinaya, and also all the participants. We had almost 65 participants for this session, and really happy to have such a high number. Thank you, ma'am.
Thank you. Thank you so much for the opportunity, and thank you all.
Okay, bye. Have a nice weekend.
Thank you.